When a drug enters the body, where does it actually go? It may remain partly in the bloodstream, enter highly perfused tissues within minutes, accumulate more slowly in other tissues, and eventually undergo metabolism and excretion. Ideally, we would measure the drug concentration continuously in every organ and tissue. In practice, this is neither possible nor necessary. We normally collect plasma samples at selected times and use the resulting concentration time curve to understand the drug's behavior. Compartment models provide the mathematical bridge between those measurable plasma concentrations and the much more complex processes occurring throughout the body. Welcome to Pharma Vault, where we turn complex pharmacological concepts into clear, structured lessons. In this video, we are going to explore pharmacokinetic compartment models, the mathematical models used to describe how drug concentrations change after administration. We will begin by asking why compartment models are needed and what the word compartment actually means. Then we will study the one-compartment model and see how its curve changes between intravenous and oral administration. After that, we will introduce the two-compartment model, including its distribution and terminal phases. We will also briefly examine the three-compartment model, compare absolute and relative bioavailability, and finish with the clinical relevance of these models. Once a drug has entered the body, it may distribute into many tissues with different blood supplies, membrane permeabilities, and affinities for the drug. However, we cannot continuously measure its concentration in every tissue. What we can measure is usually the drug concentration in plasma at several time points. These plasma measurements create a concentration time curve. We then fit a mathematical model to that curve to estimate how quickly the drug distributes and how quickly it disappears from the body. The model can also help us estimate parameters such as clearance, distribution volume, and half-life, and predict concentrations after different doses. A compartment model is therefore a useful simplification. It does not reproduce every physiological detail. Instead, it captures the features necessary to describe and predict the observed concentration time data. A pharmacokinetic compartment is a hypothetical kinetic space in which the drug is treated as having a uniform concentration at any given time. It is important to understand that the compartment is not necessarily a specific organ, anatomical structure, or body cavity. Tissues can be grouped within the same compartment when the drug demonstrates sufficiently similar concentration time behavior in those tissues. Within each compartment, drug mixing is assumed to be rapid and uniform. This is a mathematical assumption, not a statement that every drug molecule physically reaches every tissue instantaneously. The number of compartments also depends on the particular drug and the quality of the observed data. The same body may be adequately represented by one compartment for one drug and require 2 or 3 compartments for another. Therefore, compartment number describes the kinetic behavior of the drug, not the anatomical complexity of the body. In the one-compartment model, the body is represented as one kinetically uniform unit. After the drug enters systemic circulation, its distribution is assumed to occur rapidly relative to the sampling times. As a result, a separate distribution phase cannot be distinguished in the observed plasma data. At any particular time, the measured plasma concentration is assumed to represent the concentration pattern of the entire modeled compartment. Drug is then removed from this compartment through elimination. Following an intravenous bolus under first-order elimination, the plasma concentration decreases as a single exponential phase. This is called a monoexponential decline. Physiological distribution still occurs, but it is either sufficiently rapid or insufficiently resolved by the samples to appear as a separate phase. The number of compartments and the route of administration describes two different aspects of the model. The compartment structure describes what happens after the drug reaches systemic circulation. The administration route describes how the drug enters that structure. With an intravenous bolus, the entire dose enters systemic circulation immediately. There is no absorption phase, and intravenous bioavailability is 1 or 100%. The concentration therefore begins at an estimated initial value called C0 and immediately declines. With oral administration, the drug must first be liberated from its dosage form, dissolve and cross the gastrointestinal barrier before reaching systemic circulation. The plasma concentration begins near 0 and rises while the rate of absorption exceeds the rate of elimination. It reaches a maximum concentration, Cmax, at the time Tmax. After this point, elimination becomes dominant and the concentration declines. Therefore, the IV and oral curves look different even though both may be described using the same one-compartment structure. The route changes the drug input and the early curve shape. It does not necessarily change the number of compartments. For a one-compartment IV bolus model with first-order elimination, concentration at time t is described by C at time t equals C0 multiplied by e to the power of minus kt. C at time t is the plasma concentration at the selected time. C0 is the estimated concentration at time 0 immediately after the dose has entered the compartment. The letter K is the elimination rate constant and T is elapsed time. Because this is exponential elimination, the drug does not lose the same amount during every time interval. Instead, it loses the same fraction per unit time. The initial concentration is related to dose and apparent volume of distribution. C0 equals dose divided by Vd. For the same dose, a larger apparent distribution volume produces a lower initial plasma concentration because the drug appears to be distributed through a larger volume. The elimination rate constant is related to clearance and distribution volume. K equals clearance divided by Vd. Clearance describes the volume of plasma from which the drug is completely removed per unit time. The rate constant K has units of inverse time and describes fractional removal. Half-life is the time required for concentration to decrease by 50%. It equals 0.693 divided by K. Substituting clearance divided by Vd gives 0.693 multiplied by Vd divided by clearance. This relationship explains two important effects. If clearance increases while Vd remains constant, K increases and half-life decreases. If Vd increases while clearance remains constant, K decreases and half-life increases. These relationships apply under the assumption shown here: IV bolus input, one kinetically uniform compartment, and first-order elimination. For oral administration, the equation must describe two simultaneous processes: absorption into the systemic compartment and elimination from it. The equation shown here is known as the Bateman function. C at time t equals F multiplied by dose and the absorption rate constant divided by Vd multiplied by the differences between Ka and K. This is then multiplied by the difference between e to the power of minus Kt and e to the power of minus Kat. F represents bioavailability, the fraction of the administered dose reaching systemic circulation unchanged. Ka is the absorption rate constant, while K is the elimination rate constant. At time 0, the predicted systemic concentration is 0. It then rises as absorption delivers drug into the compartment. At Tmax, the rate of drug entering systemic circulation is equal to the rate of drug leaving through elimination. This produces Cmax. Tmax can be calculated as the natural logarithm of Ka divided by K divided by the difference between Ka and K. Total systemic exposure is represented by the area under the concentration time curve, or AUC. For a linear oral model, AUC equals F multiplied by dose divided by clearance. If F and clearance remain unchanged, altering Ka changes the shape of the curve but not the total AUC. Faster absorption generally produces an earlier Tmax and a higher Cmax. A slower absorption generally produces a later Tmax and a lower Cmax. This model assumes a single dose, first-order absorption and elimination, and no absorption lag time. Some drugs do not produce a single exponential decline after intravenous administration. Instead, plasma concentration decreases rapidly at first and then more slowly. This behavior can be described using a two-compartment model. The central compartment contains the sampled plasma and represents the space with which the drug equilibrates relatively rapidly. The peripheral compartment represents tissues that equilibrate with the drug more slowly. The administered dose enters the central compartment. The drug moves from central to peripheral according to K1-2 and returns according to K2-1. In this standard model, elimination occurs from the central compartment and is represented by K10. The compartments communicate continuously, so distribution is reversible. Again, these are kinetic spaces. The central compartment is not simply the blood, and the peripheral compartment is not one particular organ. The important difference from the one-compartment model is that distribution is now slow to appear as a distinct early phase in the observed plasma curve. After an IV bolus in a two-compartment model, plasma concentration is described by a biexponential equation. C at time t equals A multiplied by e to the power of minus alpha t plus b multiplied by e to the power of minus beta t. The observed concentration is therefore the sum of two exponential components. Alpha describes the faster macro-rate constant associated with the early distribution phase. During this phase, plasma concentration falls stiffly because drug is both being eliminated and moving from the central compartment into the peripheral tissues. Its distribution half-life is 0.693 divided by alpha. Beta describes the slower terminal macro-rate constant. By this later stage, distribution between the central and peripheral compartments is approaching equilibrium, and systemic drug loss becomes the dominant visible process. The terminal half-life is 0.693 divided by beta. However, beta should not automatically be called the elimination rate constant. Both alpha and beta are hybrid or macro rate constants. They depend on distribution and elimination together. A and B are mathematical coefficients that determine the contribution of each exponential component. They are back-extrapolated intercept coefficients, not the concentrations inside the central and peripheral compartments. The macro constants are connected to microconstants. Alpha plus beta equals K12 plus K21 plus K10. Alpha multiplied by beta equals K21 multiplied by K10. Usually, alpha is greater than beta, which is why the distribution phase is faster than the terminal phase. Here, both routes use the same two-compartment structure. After an IV bolus, the drug enters the central compartment immediately. The resulting curve begins at a high concentration, and the early alpha distribution phase is directly visible. This is followed by the shallower beta terminal phase. After oral administration, an additional process is introduced. The drug must first be absorbed into the central compartment according to Ka. The concentration begins near zero and rises towards Cmax at Tmax. While this is happening, drug may already be moving between the central and peripheral compartments. Consequently, the rising absorption phase may partially or completely conceal the early distribution phase. The observed oral curve therefore contains contributions from Ka, alpha, and beta, even when these phases are not visually separable. There is also an important terminal slope rule. When Ka is greater than beta, absorption finishes relatively quickly, and the latest slope usually reflects beta. When Ka is smaller than beta, absorption is slower than terminal disposition. In that situation, the late curve may be controlled by absorption rather than beta. This is called a flip-flop kinetics. Therefore, the final slope should not be interpreted automatically without considering the absorption process. Some concentration time datasets contain three distinguishable exponential phases. These may be described by a three-compartment model consisting of one central compartment and two peripheral compartments that equilibrate at different rates. The rapidly equilibrating peripheral compartment communicates with the central compartment through K12 and K21. The more slowly equilibrating peripheral compartment communicates through K13 and K31. For certain lipophilic drugs, adipose tissue may contribute to a slowly equilibrating peripheral space. However, the slow compartment should not simply be labeled adipose tissue because compartment identity remains drug dependent. After IV administration, the concentration can be described by three exponential terms. C at time t equals AE to the power of minus alpha t plus B e to the power of minus beta t plus C e to the power of minus gamma t. Alpha represents the fastest phase, beta an intermediate phase, and gamma the slow terminal phase. A three-compartment model should be used only when the data supports the additional complexity. Adding more compartments does not automatically create a better or more clinically useful model. Bioavailability is the fraction of an administered dose that reaches systemic circulation unchanged. Intravenous administration is used as the reference for absolute bioavailability because an IV dose enters systemic circulation directly and has an F of 1. Absolute bioavailability compares an extravascular route such as oral administration with IV administration. Because the doses may differ, we compare dose normalized AUC values. Absolute F equals oral or extravascular AUC multiplied by the IV dose divided by IV AUC multiplied by the extravascular dose. Absolute bioavailability may be reduced by incomplete absorption and pre-systemic loss, including metabolism in the intestinal wall and liver before the drug reaches systemic circulation. Relative bioavailability compares one non-IV test formulation with another non-IV reference formulation. Relative F equals the test AUC multiplied by the reference dose divided by the reference AUC multiplied by the test dose. Because this is a relative comparison, the result may be below, equal to, or above 1. AUC primarily describes the extent of systemic exposure. Cmax is the maximum observed plasma concentration and Tmax is the time required to reach it. These parameters provide information related to the rate of drug input, although both can also be influenced by distribution and elimination. Valid comparisons require equivalent study and correctly dose normalized AUC values. Bioavailability affects exposure, but it does not by itself determine whether the drug follows a 1, 2, or 3 compartment. Compartment models are clinically useful because they help connect dosing with predicted concentration time behavior. Model parameters may be used to simulate loading doses, maintenance regimen, and the repeated administration. The appropriate distribution volume depends on the clinical target and the time point at which that target should be reached. Sampling time is also essential. In a multi-compartment drug, a sample taken during the early distribution phase may be considerably higher than the later equilibrated plasma concentration. For example, digoxin concentrations are interpreted using samples collected after the distribution phase rather than immediately after administration. Compartment models also explain redistribution. After a single dose of some intravenous anesthetics, the effect may decrease as drug moves away from highly perfused effect site tissues into peripheral tissues. This can occur before a large proportion of the drug has been eliminated. Finally, patient characteristics can alter model parameters. Renal or hepatic impairment may reduce clearance. Edema, obesity, or altered protein binding may affect apparent distribution volume. These changes can influence peak concentration, half-life, and accumulation. The goal is not to choose the most complicated model, it is to choose the simplest model that adequately explains the observation and answers the clinical questions. Let us bring the main ideas together. First, compartments are kinetic abstractions, not fixed anatomical organs. Second, administration route changes how the drug enters the system. IV administration produces immediate systemic input, while oral administration introduces bioavailability and absorption. Third, a one-compartment IV bolus curve declines monoexponentially. A two-compartment IV bolus curve declines biexponentially, with an early alpha distribution phase and a later beta terminal phase. A three-compartment model adds another distinguishable kinetic phase, but should be used only when supported by the data. Finally, AUC allows us to evaluate systemic exposure and bioavailability, while appropriate model selection and sampling time are essential for correct clinical interpretation. The central principle is simple: use the simplest model that adequately explains the observed data. Thank you for watching. In the next Pharma Vault video, we will follow the complete LADME journey of a drug through the body: liberation, absorption, distribution, metabolism, and excretion. We will also examine phase 1 and phase 2 metabolism, as well as enzyme induction and inhibition. See you in the next video.